I welcome all the attendees in this special public lecture, which is being jointly organized by the National Academy of Sciences, India, NASI Delhi chapter, and the MHRD-Institution Innovation Council, the Dayal Upadhyaya College, University of Delhi, under the aegis of the DBT Star College program. And today we have the special public lecture on Nature's Engineers, Active Matter. It's going to be delivered by Professor Julia Yeomans, and who is associated with the Rudolf Center for Medical Physics, University of Oxford, UK. And as a customary, I'll introduce Professor Julia, who received her DPhil thesis on critical phenomena of disordered systems from Oxford University in 1979, and is Professor of Physics at University Oxford from 2002 onwards. In 2013, she was on a visiting fellowship at Newton Institute, Cambridge, and 2011, she was visiting professor at University of Kyushu, Japan, under Enhanced Education Program. She received University of Oxford Teaching Award in 2008 and was elected Fellow of the Royal Society in 2013. She has delivered P.S. Narayanan Memorial Lecture at the Department of Physics at Indian Institute of Science, Bangalore in 2017, and Annual Edward Dayal Lecture in Lincoln in 2019. She has published more than 250 publications with a huge numbers of citations and an H-index 52. And her current editorial appointments include Oxford Encyclopedia Advisory Board, from which she has been associated from 2018 onwards, Editorial Board of Journal of Physics A, Editorial Board Member, Molecular Physics, Editorial Board with Philosophy of Transaction A, an editorial board member of Physics Review E, and of Journal of Statistical Physics. She is currently interested in collective behavior of atom systems, mechanobiology, swimming at low Reynolds number, and droplets and wetting. With these words, I welcome Professor Julia, and I request her to kindly share her screen.
Okay, let's see if we can get this to work.
Yeah.
Is that okay?
Absolutely fine.
Good.
Please go ahead.
Well, thank you very much for that kind introduction. It's very, very nice to be here talking to you. Although, of course, I think it would be even nicer to be there in person. I've visited India about 3 or 4 times now, and I've always loved it. It's the most amazing country. And I hope to spend my sabbatical there this fall. But for obvious reasons, that's fallen through. But I hope it won't be long before I can spend a long time exploring Delhi and exploring the rest of India. What I'm going to talk about today is really physicists muscling in and trying to do biology. I'm going to talk about active matter, which has become a way of looking at biology from a physics point of view, and a new way of thinking about statistical physics. So what I'll do first is just tell you what active matter is, and have a really long introduction, because this is a fairly general seminar, a long introduction trying to explain why I think active matter is exciting, and the sort of questions that need to be answered. Then I'll talk about something called active turbulence, and then I'm going to talk about a parable using the ideas that I've used, the hare and the tortoise story applied to bacteria. So active matter, what is active matter? Well, it's become the name for things which take energy from their surroundings, and each individual unit of this active system takes energy from the surroundings and uses it to do work. So that's really a fancy way of saying anything that's alive, anything that's biological. Animals, us, are always taking energy from the environment in the form of food normally, and then using it to move around, to do all the things that keep us alive. That's a bit hard for physicists, so we tend to think of that on a much smaller length scale. And so we tend to think of things like cells, individual cells, or going on an even smaller length scale, molecular motors, which are the engines of the cells. But anything which is alive is always using energy and using it to do work. And normally the sort of work I'll be thinking about is motion, the way things move. So let's start at the smallest possible length scale. Let's start by thinking about molecular motors, which are the engines of the cell. And have you ever thought how our bodies work? I mean, we know about engines. If you say how do you move, you immediately think about muscles. But then how do muscles work? And in the end, it has to be worrying about how cells themselves operate together to allow us to move bits of our body, to allow us to move things around our body. So inside a cell, what are the engines? How are things moved around inside cells so that we can do all the things we need to do to live? Now, inside cells, things are pretty, there's lots of things going on in there. They're pretty crowded and they're very small. And so if you, say, wanted to move a protein from one side of a cell to another, how would this happen? Wouldn't be easy. The protein couldn't sort of swim like a little swimmer because it would be battered around by Brownian motion by all the other particles in the cells. And so it would have real problems getting from one side to the cell to the other. So how's nature solved this problem? How does it do transport on very small length scales? Very small length scales, submicron inside a cell. And this is one of my ever so favorite movies of all time. This is from a YouTube movie, Inner Life of the Cell, and it shows how things are moved around inside a cell. First of all, the cell lays down tracks called microtubules. That's so that the motors can move around without getting buffeted by their surroundings. And then these tiny little engines move along the tracks. These tiny little molecular motors are able to walk along the tracks. Let me show you that again. Here are the microtubules being laid down. And then we have molecular motors moving along the tracks. The two little feet are the molecular motors. And then whatever cargo they want to move is put into a vacuole, these big vacuoles, which follow the motors. And certainly, maybe biologists are used to this, but as a physicist, the first time I saw this, I thought, oh yeah, it can't be like that. But it is. This is really what's happening inside our cells. These things are proteins. This is a kinesin motor, and it's a protein. And the sort of length scale is maybe 20 nanometers. And it works by taking molecules of ATP from the surroundings. And the ATP binds to the back foot of the motor, and it allows that foot to become unbound from the microtubule. And then because of the shape of the protein, the back foot tends to elastically move to the front. It tends to be catapulted to the front, and then it tends to bind again to the microtubule in front, allowing the back foot to in turn gain a molecule of ATP. This is just an artist's impression, a movie, but it really is more or less like that. The only real difference is that this thing is just going plod, plod, plod along the microtubule. And in fact, it's a much more stochastic process. Sometimes it walks forwards, sometimes it walks backwards, but on average, it moves forwards. Just to show you that it's real, here are a microscopy of the same thing. Here are the two heads, and then here are the two heads at a later time as they've moved along this microtubule. And so there are lots of questions, there are lots of really interesting questions here. For a start, how does nature make the proteins that are the kinesin motors? How does it put together the right molecules to end up with this really complicated structure. And then there's also a big question about control theory. How does it decide which motors are going where? How does it decide where to lay down the microtubules in the cell? And how does it decide what to pack in the vacuoles for the motors to pull around? And I really think that we don't understand any of that at the moment. We really don't understand the sort of control system in these cells, which gets things to the right places at the right times. Here's another of these motors. I'm going up slightly in length scales here. These are bacteria, and they're typically 10 microns in length, and they swim around. And the way they swim around is by their tails turning. As the tails turn, they wave and they push the bacteria along. And here is the motor which makes them turn. The motor sits across the cell membrane between the bacterial body and the tail. And the motor looks something like this. Here's the cell membrane. Here's the motor. Each of these little sort of blobs is a different protein. And it's a rotary motor. It turns. It's driven by a flux of protons which move across the cell membrane. And then here's the tail. Here's the flagella sitting out beyond the cell. Look at the length scales. This is about 30 nanometers. It's tiny. And look at the complexity. In order to work, these proteins have to be in exactly the right place. And I should say that this really is a guess. We really don't know exactly what this motor looks like, although we've got some idea. This thing can go about 100 revs per second when it's unloaded. And so it's a pretty amazing machine. If we tried to make anything like that out of metal or some sort of nanotechnology, friction would kill you very quickly. And so the cell has to be able to both make something like this and also repair something like this. So how might it do it? How might it put things together to form a motor like this? And again, this is a movie lifted from YouTube, showing how gradually one could imagine building up this sort of motor, but we really don't know how it's done. And so this really is the first reason for studying active matter. Nature is really, really good at producing tiny motors. These tiny motors basically are responsible for all the things that living creatures do. And if we could emulate what nature does, both in terms of building the motors and in controlling the motors, then we would have a whole new source of devices, which presumably would be incredibly useful and incredibly efficient. So how far have people got with building tiny motors, with using this active matter, with trying to emulate living systems? And the answer is really not very far. So let me show you a couple of recent experiments, one which shows the sort of motors that people are building, the sort of tiny motors, and one which shows self-assembly. And they really are very clever experiments, but they're nowhere near what nature can do. These are tiny gears, about 50 microns across. And those dots that you can see are tiny little swimmers, tiny little bacteria. And the bacteria are pushing into the motors and they're turning them around. It's not perpetual motion. The energy source is basically the food which is powering these bacteria. And you can get them to move, but they're not really very complicated. And certainly they're much bigger than these tiny motors that we're seeing in nature. The next picture shows a system of active colloids. These are colloids that people have tried to make active, so that instead of just doing Brownian motion, they have directed motion. The way it's done is by having a colloid. Colloids are tiny micron-sized particles, and having a blob of hematite stuck on the colloids, and then putting them in a solution with hydrogen peroxide. If you shine light on this solution, what happens is that the hydrogen peroxide reacts with the hematite, and these things act like tiny rockets, and they're self-propelled in the direction away from this blob. And if they're self-propelled, what they do is that you get self-assembly into lattices. And that sort of makes sense because these things are moving in a certain direction. They tend to hit each other and they tend to form these lattice structures. And just to check, in a minute we'll turn the light off. And then what happens is that you just get Brownian motion and the lattice structures break up. So people are starting to make tiny motors, they're starting to look at self-assembly, but there's a long way to go before we actually understand what's happening in the real systems. So why else might we want to look at active matter? That was for the engineers. Now for the physicists, we're all used to, I think, what happens if you have a large number of particles. A large number of particles, like the gas in a room, they come to thermodynamic equilibrium, and that equilibrium is described by the Boltzmann equation. What happens if you take a load of active particles and put them together? Would you get Boltzmann statistics? Will they come to equilibrium? And the answer is very definitely no, because active particles are out of equilibrium, and that's good. They're meant to be out of equilibrium. Anything alive is out of equilibrium because it's taking in energy the whole time and it's using it to do work. Because it's really bad if living things come to thermodynamic equilibrium. So we've got a new playground. We've got a playground where when we put together loads of active particles, we end up with a system where you can look at the collective behavior of a system out of equilibrium. So let's see what happens. Okay, let's take loads of bacteria and put them together and have a look. So this is a dense suspension of microswimmers swimming in a film, so it's two-dimensional, so you can see what happens. These are E. coli, they're tiny swimmers, about 10 microns long. And what happens is that they swim like this. You end up with something which looks turbulent, where you get lots of swirls and lots of complicated flow patterns. And that's actually a bit strange because these things are tiny, and tiny means low Reynolds number, and low Reynolds number means flows on small scales, which are usually very smooth. So people were quite surprised when they saw this. Let's look at another system. Okay, let's go back. Let me use the excuse to show you this yet again, this movie, because it's formed the basis, these microtubules and these kinesin motors. So remember, these are the microtubules, long polymers, and these are the little molecular motors. They form the basis of one of the most important experimental systems for dense active matter. There was some beautiful work about nearly 10 years ago now from Zorimer Dodgik's group, then at Brandeis, where he isolated the microtubules, actually from the brains of cows from the abattoir in Boston. So look away now if you're vegetarian. It's a bit gruesome, this bit. Okay, so these are these microtubules. And then he put them basically in a suspension with loads of special sorts of molecular motors, special sorts of kinesin motors. And the motors were special because they have two heads. So they're two-headed motors, and so they can form bridges between the microtubules. These motors are directional. They can only walk in one direction along the microtubules. And so if these guys are walking downwards and these guys are walking upwards, what will happen is that the microtubules will get pushed relative to each other. And you have a collective active system of lots of microtubules, lots of these long polymers being driven around by the kinesin motors. So let's have a look and see that system in action. What you'll see is the little white threads are basically bundles of these microtubules, and the motors are pushing them relative to each other. So what happens is that they buckle, and again you get this chaotic sort of flow state. Let's play that again. Chaotic sort of flow state, but it's not just ordinary chaos, right? You get these sort of structures appearing, these sort of gaps and these sort of bends as the microtubules get folded up and buckle. So then there's the question, is this active turbulence the same in this microtubule motor system as it was for the bacteria? The nice thing about this active turbulence is it gives me an excuse to show fun movies. This is another active system. This is a bit different because the active system is much bigger. They're fish, they're much more intelligent animals, and so you could imagine some sort of signaling between them. But again, you get patterns. The thing attacking them apparently is a seal, as I got told when I gave this lecture in Denmark. You can see it's not a shark because of what the surfboarders don't do. But this seal is obviously causing some sort of pattern formation in the fish school. And we don't understand how this happens. We don't understand whether it's just physics or whether you need some sort of biological signaling as well. And of course, the other very beautiful one, where you have pattern formation with large number of active particles, here macroscopic active birds moving together, is that you end up with this sort of beautiful patterns where you get starlings. And just about 3 or 4 miles away from where I live, this happens every November. And if you can bear the cold, it's lovely to go out and watch them. So we'd like to understand this sort of pattern. And so a second reason for looking at active material, and in particular active systems where you get a large number of particles together, is to understand non-equilibrium statistical physics. Really, for the first time, this is giving us a handle on experimental systems where we can look at non-equilibrium statistical physics properties. One more reason is very much related to biology. Another example of active turbulence, this is active turbulence in eukaryotic cells. This is a layer of cells, very much like the cells which line your stomach and which line your lungs. And these have been taken out and put on a Petri dish, and then it's watched how they move. And you can see again, you get this sort of turbulent-like behavior. And indeed, they start moving around in a circle. And often these active systems do have this spontaneous rotation, if you can find them. So does mechanics have anything to do with processes, with collective behavior and biology? We think it does, but the jury is still out how much it's chemical signaling and how much it's mechanical signaling. But let me show you 2 examples of really amazing biological processes where probably physics and the collective behavior of active systems has something to do with explaining it. I say something to do because we don't know yet. but see what you think. This is Drosophila, fruit fly, and it's how a Drosophila changes from being a tiny embryo to start looking like a fruit fly. So these are individual cells that you're seeing here, and this is how the Drosophila evolves in time. You can see the length scale is about, this scale bar down here was about 50 microns. And in particular, flows are important. Collective behavior is important, and flows are important in this system as you get the thing segmenting so that the different areas of its body start to be distinguished. Traditionally, this has all been put down to chemistry. Now people are starting thinking about it in terms of mechanics and active matter. Another one which is nice, which I learned about recently, comes from a lab in Chicago. And again, Drosophila, the fruit fly. This is an egg chamber of this fruit fly. And the egg chamber here has the eggs inside, and it's lined by a layer of cells, a layer of these eukaryotic cells. And the cells don't just sit there. For some reason, they start rotating, very like we saw the cells rotating in the slide I showed you just a couple of slides ago. Here's a movie which labels some of the cells, and you can see them rotating together around the outside of the egg sac. The speculation is that the rotation helps elongate the egg sac, which starts off as a circle and ends up as an ellipsoid. Some of the beautiful experiments that have been done in Sally's lab is that the cells were sent to sleep by adding a chemical, basically a sleeping pill for cells, and then the rotation stopped. When she washed out the chemical, the rotation started again, but the cells started moving in the opposite direction. And if this is repeated, if you stop the cells using some sort of chemical which stops them moving but doesn't harm them, and then wash out the chemical so they start moving again, they will move in the opposite direction, and this will keep going each time you stop them. They change direction. That's very strange collective behavior, and we don't understand why it's happening. And I think it's a very interesting question why it might be happening. Okay, so that was really a long introduction. That explains why I care about active matter and how I think there's some really, really exciting questions to be answered. So now let me just turn to one of those questions, which is the one we've been thinking about over the past few years. And of course, when you actually do things you can actually do rather than things you'd like to do, it's perhaps less wide-ranging, but I think we've made some progress. And where I want to end up is to try and convince you that topological defects are important in biology. So first of all, I need to talk about some sort of background. I need to just tell you a bit about liquid crystals. And then I'll say a little bit about swimming at low Reynolds number, because that will give us the background physics to be able to understand active turbulence and introduce motile topological defects. So first of all, liquid crystals. Liquid crystals are things which are shaped like this. So either a molecule, which is this shape, or something, a substance, a nematic has lots of molecules which are long and thin like this, or you could imagine a collection of colloids which are shaped in a long and thin way, which have a special axis, which is called the nematic axis. If you take a system like this, and you put the colloids together, or you put the molecules together, often they will tend to line up so they're all pointing in the same direction. This gives them more room, it lowers their entropy, and you end up with something called a nematic phase. The pneumatic state is really important because this is why I can talk to you now and you can see what's going on. It's a pneumatic which is in your laptop screens because the special direction of the molecules interacts with light and allows the screens to be light or dark. Displays are based on the technology of these pneumatics. Okay, so pneumatics are like this. Often at high temperatures, you'll have them disordered. This is my favorite pneumatic from Oxford. This is just outside my college window. Gets pretty noisy in the summer. High temperatures in the summer, everything's disordered, but at low temperatures in the winter, everything lines up and you get this nice pneumatic phase, which has a special direction to it. So the really important thing about a pneumatic state, which is going to be relevant to what I'm going to be talking about here, is that you can get defects in the pneumatic order. Here the thing is trying to line up so the pneumatic direction is horizontal, but you can get places where this goes wrong. And these places are called topological defects. They're called topological defects because they sort of act as knots. You can't unwind them by locally changing the director field. You have to change it all the way out to infinity. So actually, a little defect like this, little defect like this mucks up the director field, the direction of this pneumatic, all the way out to infinity, and it has infinite energy. So these are very special sorts of defects called topological defects. If you have a two-dimensional pneumatic, which is what we're going to be worrying about here, the most important defects are the +1/2 ones, which look like this red one here, look like a comet, and -1/2 ones, which look like this, which have this threefold symmetry. Usually what happens is that they appear in pairs, because if they're together like charges, they don't have infinite energy, and then they gradually move towards each other and anneal out in pairs to leave a perfect pneumatic. So please can you remember these shapes? This is going to come up again, this +1/2 defect looking like a comet, and the -1/2 defect with a threefold symmetry. So that's the first thing. Then the second sort of bit of background is low Reynolds number hydrodynamics. Those of you that are used to the Navier-Stokes equation will know that fluids flow in a certain way, which is very different at large length scales and small length scales. At small length scales, or equivalently high viscosities, normally the way fluids flow is very smoothly. And what I'm showing you here is a movie which demonstrates the main ideas of hydrodynamics at low Reynolds number, at hydrodynamics, how fluids flow at very small length scales. So what these guys are doing is putting dye into a beaker, and I know it's not small length scales, but it is high viscosities which behaves in the same way. Then got a pair of tame graduate students to hold the beaker down, and then mixing up the dye in the fluid. And maybe you could count the number of times they go round. I think we're up to 3 now, and I think it goes on till this thing's gone round 7 times. Now, this is quite a famous movie, so I apologize for those of you that have seen it before, but it really is a very, very beautiful demonstration of how hydrodynamics works at this low Reynolds number limit, how it works on small length scales. So now we're going back, we're unwinding this fluid. And of course, what you expect is, I mean, normally if you mix something up, it doesn't unmix itself. doing it quite slowly. And one more time round. And what we see is you get back to where you started from. This, more than anything, says what happens in fluid mechanics at small length scales at low Reynolds number. Everything is reversible. If you do something to the fluid and then do the other thing backwards in time, then just reverse what you did in time, you get back to where you started. Now, bacteria and active particles that swim do so on these small length scales. And so there are two important things which are a result of this. The first is something called the Scallop theorem, which tells you how these tiny bacteria swim. Because any swimming stroke, which is the same forwards and backwards in time, will move you. It might move you, but then it will move you back to where you started from. So a swimming stroke, which is not time reversal, non-invariant, which is not different forwards and backwards in time, the thing might move, but then if it carries on with its stroke, it will go back to its original position. So bacteria have to propel themselves with a swimming stroke which is not invariant under time reversal. The way it does that is to have tails like this, where waves go down the tails. Waves have a direction in time, and so by having these long tails, the bacteria has a swimming stroke which looks different forward and backwards in time. The wave along the tail would seem to change direction, and so that's what allows it to swim. So these funny shaped bacteria are just trying to overcome the low Reynolds number hydrodynamics they have to live with. The other special thing, which is more relevant to us in fact, is to look at the shape of the flow fields that bacteria produce. If you swim in the swimming pool or wriggle about in the bath, you're going to set up flow fields around you. Similarly, when bacteria move, they set up flow fields. They make the fluid around them move. And these are some very lovely experiments from Cambridge, which show the shape of the flow field they set up. And you can see that it's a very special shape. What happens is the bacteria tend to push fluid out from their front and their back and pull it in from the sides. And so what happens is that they produce these flow fields, and it's actually the shape of these flow fields which is important in understanding active turbulence. So let's go for it now. Let's look at active turbulence and try and understand what's going on. Here's the active turbulence. Let me just show you again what we're trying to understand. We've said that at low Reynolds numbers everything's very smooth, but this certainly isn't very smooth. And so we've got to explain why we get this sort of flow pattern. And another way of looking at it is by plotting the vorticity field. What I've plotted here is the circulation of the fluid, how it's moving round. So blue means that it's moving round in a circle clockwise, and the red shows you that you're moving anti-clockwise. You're getting flow vortices set up. And typically the scale of these flow vortices is maybe, what's that going to be? About 50 microns, about 10 times the size of a bacterium. So you're getting these collective vortices set up, and that's very special, a very special feature of this active turbulence. And the reason you get it is that each of these little bacteria is producing one of these flow fields with special symmetry. And what you can show is that if you start off with bacteria trying to swim in a nice organized way, so they're all moving like soldiers in the same direction, any tiny fluctuation means that when you add up the flow fields they produce, it works, it flows in the direction of these arrows. And so what's going to happen is that that tiny perturbation is just going to grow and grow. And the nice, smooth swimming is going to be broken up. To show you that again, what we did is we wrote down the equations which describe this. They're sort of like the Navier-Stokes equations, but rather more complicated to take account of the flow fields of the bacteria. And then we plotted the vorticity field, and it looks like it's meant to look like. And then if you sort of zoom in on this, so you can see the individual swimmers, the individual bacteria, you can see that they look like one of these pneumatics. And what's happening is that the flows are causing these bends in the pneumatic order. And similarly, the bends in the pneumatic order are causing the flows. And so it's because you get each of these bacteria producing stresses in the fluid, producing flows in the fluid, that you end up with the active turbulence. So let's look now at, I'm going to miss this bit out. Let's look at these simulations and let's say, remember I was talking about this being a pneumatic. Okay, I was talking about the bacteria acting like one of these pneumatic things. So you'd expect to see topological defects. Remember, we had these +1/2 and -1/2 topological defects. Let's look in the simulations and see how the +1/2 defects and the -1/2 defects move. And what you'll see is that they annihilate each other in pairs, just like charges, which is what you would expect. But then by the end of the simulation, there seem to be the same number as they were at the beginning, more or less. So let's play it again and see. Whoops, let's try and play the right thing again. Let's play it again and see what's going on. And what you see, if you look carefully, I still have trouble finding them, having looked at this for ages, is that they destroy each other in pairs, but also if you look up here, they pop out in pairs. The topological defects are not just destroyed in pairs, but because this system is active, because it's always taking energy from its surroundings, it has the energy to create these topological defects. And so what's very special about this active turbulence is that you get a sort of gas, a background gas of +1/2 and -1/2 topological defects which appear in the system. Let's have a look at that in a real system. And rather than looking at the bacteria where they're a bit hard to see, I'm going to look at the microtubule motor mixtures. And you can see these shapes here. And actually, if you get used to looking at it, this is a +1/2 topological defect. And this one here is a -1/2 topological defect. It's really hard to see till you get used to it. So let's look at that in a still. Here are the microtubules being bent by the molecular motors. And as they're bent, what you find is that you create defects here, a +1/2 defect and a -1/2 defect. And the +1/2 defect moves away from the -1/2 defect. Okay, so active turbulence. The state that you see in these active particles when you put lots of them together is a state where you have topological defects. At least in this synthetic biology system, you're seeing topological defects. Let's now see if we can see them in a real biological system. So the last thing I want to do is tell you the story of the hare and the tortoise. Now, I did look up to see if it was on the web to see if you'd heard of the hare and the tortoise parable in India, and I think it's pretty much a global thing, so I hope you know what I'm talking about when I talk about the hare and the tortoise. Here we are. And this is work which will come out in Nature Physics in the next couple of weeks. Well, shortly, I'm not sure exactly when, it's just been accepted by Nature Physics. And here are the people I've collaborated with in this. And in particular, many thanks to Armin, who's now in Denmark, who was a postdoc with me at the time, who did the theory, and Ollie Meacock, who did some beautiful experiments. So I'm talking about bacteria again now, but they're a little bit different from my swimming bacteria. These are twitching bacteria. They're Pseudomonas, and they move around on surfaces. So here they are busy twitching away. And you can see they look like these nematics, right? They're long and thin, and you don't get global nematic order, but locally they tend to line up, and you tend to get nematic order. So they're behaving like one of these active nematics. And the question that my colleagues wanted to ask in zoology is what happens when you have two different sorts of bacteria mixed together? And in particular, two different sorts of bacteria that move at different speeds. So what they did is they took the wild type bacteria, the wild type Pseudomonas. Okay. I always used to think the wild type meant ones they got out of a pond, and it sort of does really. It means ones that they haven't changed in any way. So this would be a sort of bog standard Pseudomonas. And then they genetically altered some of these things. So they had more of these little pili, more of these little feelers, and these are the feelers which they use to push themselves around. So these guys, these guys move faster than those. So we had a mixture of fast bacteria and slow bacteria. And the question was, how does it work that the fast bacteria are better at spreading out than the slow bacteria? So what this is let's just look at how these things spread. So the ones at the top are the wild type, the slow ones. They're going to move from left to right. I won't show you that yet. The ones in the middle are the fast ones, and they're going to spread from left to right. And then the ones at the bottom, the ones where basically the feet have been removed, so they're not meant to move at all. Okay, so slow at the top, faster in the middle. So let's have a look how these spread out with time. Okay, so this is the hare and the tortoise story. Remember, the ones at the top were the slow ones. Let's just play that again, if I can get it to work. Okay, ones at the top spreading from left to right. Top of the slow ones, ones in the middle are the fast ones. For some reason, these slow bacteria are better at spreading than the ones that move faster. Why? Just to check, this is the front of the colony, now of a mixed colony, where the yellow ones are the fast ones and the blue ones are the slow ones. At early time, the edge of the colony is indeed dominated by the fast yellow ones, but at later times, the yellow ones seem to have got lost, and the front of the colony is dominated by the slow ones. And it's important to say that these things don't divide very much. It's not that the blue ones are better at dividing. This is really just them spreading out due to motion rather than due to division. Okay, let's just jump back and go back now to ask about topological defects in these systems. Let's look for these topological defects. Here's the comet, and here's this threefold trefoil. And because this is a nematic-like thing, you can identify locally comet-like defects and trefoil-like defects. Here are these defects sitting there in the experimental system. Certainly you get topological defects there, right? And we knew we'd got those in our theories. I've shown you this before. Here we've got defects again. We can look at topological defects, but You might argue that, these are equations we're solving. Is this anything to do with what's happening in the real systems? And so we thought maybe we should look at a model which is closer to the bacteria. So we just had a load of rods moving around in the computer, each subject to a constant driving force and not able to overlap. And we looked at topological defects. These red ones are the plus a half defects, and those threefold ones are the minus a half defects, and they work in exactly the same way. Okay, so that's just a picture. Are these things, what can we measure to make sure the defects are the same as each other? So what we did is we measured the flow field around the defects. Just as bacteria set up flow fields, set up velocity fields, these defects will set up velocity fields. And these are the continuum simulations. These are these rod simulations. These are the experiments. This is the flow field around a minus a half defect. You can see 6 vortices. See it again in the rod simulations, but it's more noisy because these simulations are more noisy. And here in the experiments, you can see 6 vortices. Around a plus a half defect, you expect the flow field to look like this. Looks the same in the rod simulations. Looks the same pretty much in the experiments. And so we thought, we're seeing the same idea, the same sort of topological defects in all these systems. So what? Yeah, I've said we've got topological defects. I've said that these slow bacteria are moving faster than they ought to. Let's put the two together. This is a picture where the slow bacteria are picked out, sorry, the fast bacteria are picked out in yellow, and we're going to go through it twice, which is why there's a jump in the middle. And what you see is that the fast bacteria form clumps which get stuck. The fast bacteria form clumps, which you can see as yellow fluorescence, which get stuck. So what's happening? Let me tell you what's happening and then show you the evidence. What's happening is that 2 minus a half defects move towards each other. They circle around each other, and as they circle around, they set up flows and forces which push the intervening bacteria, which push the bacteria, so they're pointing out of the surface. So they're standing on their tails and pointing perpendicular to the surface. Perhaps this is the best way to see it. They end up pointing perpendicular to the surface. If the bacteria are moving slowly, these defects are moving slowly, and there really isn't enough energy to make things stand up. They just fall down again. If the bacteria are moving quickly, there's loads of energy in these defects. The defects are moving quickly, and they produce large flows, large forces. Things stand up, and they stay stood up. Let's see that really happening in a simulation. Here, things are moving slowly. The bacteria stand on their ends, and they fall down again. Here, the defects are moving quickly. The bacteria stand on their ends, and if we had a big enough simulation box, this blue region would just get bigger and bigger. You can see it in a larger simulation. The defects are moving slowly, nothing happens. Here they're moving more quickly, and you sort of start to get regions where things are standing up. Here you get large regions where things are standing up. So the story is that the fast bacteria form these domains where they're standing up and get stuck, allowing the slow bacteria to be more efficient in spreading. I've shown you simulations because it's perhaps easier to see, But the really clever thing is Ollie's experiments, which show the same thing. They don't perhaps look so amazing, but I mean, they really are. To be able to catch this actually happening, what you'll see is these two orange defects move near each other, and the bacteria here stand up on end. This was like looking for a needle in a haystack. Bit tricky to see. What we're seeing is this thing here. Here you're seeing a top-down view. Here you're seeing a sideways view. Here are these yellow fast bacteria getting stuck in regions where they're standing up. So just to check, let's do a simulation with two sorts of bacteria in there. Here are the domains forming, and we counted the number of fast bacteria in the domains, and they definitely get stuck preferentially. So at least in this system, topological defects are playing a real biological role in how things work. And maybe, as zoologist friends like to say, this is relevant to evolution, that things have evolved to move at a certain speed, because if they move too fast, what would happen is that they would end up with topological defects, making them stand up on end and get stuck, so they can't spread out. Here's a summary of what I was saying. And this is just one example of several where people have now found topological defects being relevant in biological systems. I thought I'd finish just by sorry, I thought I'd finish just by showing you this thing in action with more macroscopic active systems. It shows, yeah. So anyway, I'm going to stop there. Thank you very much for listening. It's very strange giving a seminar to a computer where there's no feedback, but I'd be now very, very pleased to answer questions.
Thank you very much, Julia. I really like the last one. So there are a few questions, and I'll take up those questions one by one. So the first one is, can you explain why any small fluctuations from perfect nematic alignment cause shear flows that enhance the fluctuations?
I mean, no, really, because you have to do the maths to see that it works like that. It works because of the particular shape of the flow field around these systems. The flow field itself has nematic symmetry. You can put that flow field into the equations, and you can do a linear stability analysis on the equations, first done actually by an Indian scientist, Ramaswamy, and you can show that as a result, these systems are unstable to bend perturbations, but you really have to do the maths to be able to see it.
Okay. I'll take up the next one. Is monolayer dynamics dictated by simple rules of interaction at cellular levels, which could be involved in morphogenesis?
That's a great question. That's what we're trying to find out. Certainly, mechanics is not the whole story. There is chemistry in there. Even if you just think in terms of forces, something has to provide those forces, and that's going to depend on chemical signaling between the cells. But the jury's still out at the extent of which you can think of these things in generic physical principles, and where you have to think about sort of individual biology to understand what's going to go on.
Okay.
And the answer is going to be both, but we don't know how much of one or the other at the moment.
Okay. So I'll take up the next one. Can you explain how the dynamic filament cross-linking and sliding and excluded volume interactions promote formation of bundles at small densities?
At small densities, you're going to tend not to get bundles. In order to, I assume you're thinking about microtubule motor mixtures here. And one of the problems was actually making these things dense enough so they did show active turbulence. One has to add PEG and push them together, and so that there is enough of an interaction in order to actually see these bundles, which can then be moved around by the motors.
Okay. I'll take up the next one. How does motor proteins ensure that there is a net motion in some direction along the microtubule?
There's a symmetry breaking, which is basically a chemical one in terms of the interactions between the protein and the underlying microtubule. These molecules themselves have a directionality to them, which means that the thing is precessive in a certain direction. But it doesn't always get it right. This nice hand-over-hand walking in the movie it's not quite like that. It's not quite so efficient.
Okay. I'll take up the next one. Is the active matter could also be traced in neoplastic tissue detection via light sheet microscopy?
The picture I showed you of Drosophila is a light sheet microscopy. And I'm sorry, I don't really know more than that because I'm not an experimentalist. But it's very, very, one of the reasons that active matter has become, I think, very exciting lately is because of the very good experiments that are now becoming possible, which means you can actually test things.
Okay. The next one, can you explain why the internal friction emerges as the sole source of dissipation for ciliary oscillations?
So we're talking about cilia now.
Yeah.
Yeah. I don't see why it does. I would have thought there was viscous dissipation as well. So I think I'd have to talk to whoever asked that question to be able to make sense of that.
Okay. So what happens when some bacteria allow to move in the slight warm and water? Will there be any change in nematics?
Oh, if you change the temperature of the water?
Yes.
I suspect that the main effect there would not be to change the pneumatic ordering. The reason is that these pneumatics are a little bit different from molecular pneumatics, which certainly do react to temperature. What makes them pneumatic is really the concentration. So as you push more and more of them closer together, they're going to tend to line up. And you end up with something which is called a lyotropic liquid crystal, where concentration rather than temperature is the variable. Temperature will just be a matter of how efficient some of the chemical reactions are and whether the things can stay alive, I think, at certain temperatures. Things don't like to be, bacteria are pretty keen on staying at room temperature.
Okay. So the next one is how one can estimate or detect the biomechanism of a particular cell by active matter.
Not sure what someone's getting at there. Tell me again what the question was.
I'll repeat. In one of your statements, you have said that the active matter are out of equilibrium.
Yeah.
Isn't this feature is one of the demerit? If is it, then how one can estimate or detect the biomechanism of a particular cell by active matter?
Oh, okay.
Yeah.
I mean, this is a very interesting question, actually. We don't know the answer. Certainly you would expect, if you look at fluctuations in the cell, I mean, normally you can measure temperature by looking at the Boltzmann distribution, say, of the fluctuations in velocity. You could try and do the same thing for a cell, and people have certainly done that, and you get a non-Boltzmann-like distribution. But really there isn't a theory at the moment that is able to understand what you expect that fluctuation spectrum to look like. The idea of temperature,
Okay.
which measures the shape of a Boltzmann distribution, no longer exists. So that's something people are trying to do, but still don't really understand how to interpret what they measure.
Okay. Okay. The next one is, when we talk about the RE number in liquid crystals, how low are these numbers?
Okay, for the swimmers, the Reynolds number for a single swimmer is probably about 10 to the minus 5. If you have a lot of swimmers, then there's some argument about exactly what length scale you should use in the Reynolds number and whether the interactions matter. But you could imagine the Reynolds number going up a bit, maybe to 10 to the minus 2, 10 to the minus 3. But exactly what Reynolds number means in those situations is a bit questionable because you have other length scales in the problem.
Okay. I'll take up the last 3 questions now. Is entropy production and fluctuation theorem in active matter are expected to be different from that of regular matter?
Yes, people are doing a lot of work on simple models trying to understand that. In particular, it's not obvious how to define pressure because the thermodynamic and mechanical definition of pressure are not always the same as each other. And the force exerted on a wall by particles depends on the structure of the wall, not just on the sort of local pressure. So very different and very much a topic of current research.
Okay. Is entropy production, in regular fluid dynamics, 2-dimensional and 3-dimensional turbulences are very different. Does one expect something similar for active turbulence also?
It looks like in active turbulence, 2 and 3D are rather similar to each other. What changes is the point-like topological defects I was talking about are changed into lines, lines which correspond to topological defects, and that makes a difference. But the question is the question is quite right that inertial turbulence at high Reynolds number is really very different from this low Reynolds number turbulence. You don't see anything like Kolmogorov scaling. In high Reynolds number turbulence, you have energy cascades from large to small length scales. And in active turbulence, you really have everything pumped in at a given length scale, the scale of the vortices, and then you don't see these cascades or scaling.
Okay. So there's just 2 questions. So the first one is, can speed at which the topological defects evolve and move around be theoretically predicted? And in particular, based on what inputs, for example, cell division speed?
Okay, cell division is not really what you need. What you need is the interactions between the different cells. For simple models, yes, we can work out the velocity field around one of these defects. By simple models, I mean a sort of souped-up Navier-Stokes type level story. What we don't understand is exactly how to go from the models to the experimental systems like the cells. So I think we could estimate the defect speed, but not really calculate it for a real system.
Okay, so I'll take up the last one. In active turbulence, where there are topological defects formation and annihilation, at any given instant, is there any condition that the total charge of defects is zero?
Absolutely, yes. You can only have them created in plus and minus pairs and destroyed in plus or minus pairs. Just from that's a sort of topological rule, absolutely fixed topological rule.
Okay, okay. Thank you very much, Professor Julia. I hope you must have also enjoyed the questions which have been put up by the attendees. And this has been a really wonderful session where there has been a lot of new opportunities, new areas which have opened up to people who are working in the different like life sciences, engineering, biosciences, and all of them. So any concluding remarks from your side, then I'll close the session in that case.
Well, no, just thank you very much, and thank you to everybody for listening.
Sure. Thank you. Thank you very much. So I
Okay.
thank all the attendees, and I thank Professor Julia on behalf of the National Academy of Sciences India Delhi chapter and the Indian Upadhyaya College for sparing her valuable time today on August 14th for delivering a special public lecture. Thank you very much.